Good evening, everybody. We'll start the webinar. Good evening. I would like to welcome you all to the webinar on future trends in forensic science, the art and science of forensic investigations, unveiling the secrets of crime solving on the 11th of July, organized by BioLeaks, a non-profit organization which is a globally recognized professional association that operates under the TechnoRate Group. It serves to propel and fuel all innovative works of research with immense potential in the fields of healthcare, life sciences, pharmaceutical sciences, and medical sciences in general. In this capacity, BioLeaks has been directly responsible for a significant amount of revolutionary developments that have taken place in these fields over the past few decades. Aside from pushing the boundaries of innovation and discoveries in these disciplines, BioLeaks also has a program in place to sustain development so that the pace of progress doesn't just increase constantly, but also remains steadfast throughout. Now, coming to the webinar, BioLeaks is organizing a free webinar on the topic Future Trends in Forensic Science with the theme The Art and Science of Forensic Investigations, Unveiling the Secrets of Crime Solving on July 11th, 2023 from 5 p.m. to 6 p.m. Forensic science is a multidisciplinary field that includes various scientific disciplines to investigate and analyze evidence in criminal investigations. It plays a crucial role in solving crimes by applying scientific principles and techniques to collect, preserve, and analyze physical and digital evidence. Forensic science covers a wide range of specialized areas, including forensic DNA analysis, ballistics, fingerprint analysis, forensic anthropology, toxicology, digital forensics, forensic odontology, and many more. These disciplines utilize advanced technologies and methodologies to examine and interpret evidences, reconstruct crime scenes, identify perpetrators, and establish a link between suspects and the crime. I now welcome the keynote speakers of today's webinar. The first keynote speaker is Ms. Aishwarya MV, Assistant Professor, Department of Forensic Science, Dhanalakshmi Srinivasan College of Arts and Science for Women, Tamil Nadu. And the second keynote speaker is Dr. Arjun Rao I, Assistant Professor, Department of Life Sciences, Christ University, Bangalore. So in the meeting, if anybody has any questions, please mention it in the chat box. So after the lecture, the speakers will answer your questions. Now, I would like to welcome Ms. Aishwarya MV. Aishwarya MV is an Assistant Professor of Forensic Science in the Department of Forensic Science, Dhanalakshmi Srinivasan College of Arts and Science for Women. She has done her internships from organizations like Kerala Police Academy, Haryana Cyber Police, and Karnataka Forest Department. She received her MSc in Forensic Science from Jain Deemed University, Bangalore. She has 2 international publications to her name. Her area of expertise includes digital forensics, cyber security, and forensic psychology. I welcome you, ma'am. Ma'am, are you there? I request you to start your presentation.
Hello.
She'll start her presentation in a few minutes.
Am I audible?
Yes, yes.
Hello.
You can start.
Hello. Yeah. So, can you please share the screen?
Yes, the presentation is ready.
Yeah. So again, good evening to one and all who is here. I'm myself again introducing Aishwarya, and thank you so much, Aljos, for giving me a brief introduction. So now we'll start with the basics. So we all know that you all are from the case of forensic science and where this forensic science name emerged, how the forensic science had developed, how it has been going on. These are the three fundamental things which we are.
Ma'am, you're not audible. Can you listen? Can you hear me?
Now?
Yes, yes. You are audible. You're audible. You may
Hello.
start.
Yeah.
Yes, yes, I can hear you.
So here is forensic science. So start with forensic science. So before going to study what is forensic science, we all have watched, all kind of CBI movies or, all kind of CID movies and kind of stuffs we had watched, right? So what is forensic science? Forensic science is what? It's known as application of science. What is application of science? Something related to science that we know. But when the science has been related with the law, what is law? Law can be anything. Yes or no? In the case of cyber law or any kind of law which is applicable in the court of, this is known as. So forensic science, it's an application of science as well as law. This is known as what? Forensic science. And also within this forensic science,
Ma'am, I think your internet connectivity is a bit low. We're not able to hear you.
so actually I'm having this net range here because now are you hearing me?
Yes, yes, I can hear you. You may
Hello.
continue. Yes, yes, I can hear you.
Yeah. If I'm not audible, please let me know, okay, sir? Yeah. Okay, so now what is forensic science we told? It's an application of science relates with the law. So can you change the slide? Hello? Hello?
Yes, yes, I can hear you. You may continue.
Hello? Yeah. So now we know that in the case of forensic science, we have n number of applications. So whenever you're studying forensic science, we have forensic chemistry, forensic biology, forensic physics, geology, and n number of things we are having. And before going to all this kind of things, we have to know what is forensic science. So as I told already, it's an application of science as well as law, right? Now we're going to study the Edmund Lockhart. Now we'll be having a doubt like who is Edmund Lockhart? Edmund Lockhart is not the one person who is known as the father of modern forensic science. Okay, father of modern forensic science is called as Edmund. So this Lockhart, he used to tell like whenever there comes a contact, it leaves a trace. Means, say for example, if you're touching your hand with an in table, okay, so what will happen? Your impression will be there, right? So this is what he told. Whenever you touches a thing or another thing, your impression will be there. Even if it is anything, no issue, but there leaves an impression. Okay, so Edmund Lockhart is the father of modern forensic science. Whenever you're learning about or whenever you're getting to know, about forensic science, n number of things will be coming to your mind. Say, for example, the CID movies or, any kind of series which are seeing Stranger Things, say, for example, anything, everything will be coming. But see, students, there are, a more number of backstories behind the term forensic science. Okay. So Edmund Lockhart, he is known as the father of modern toxicology, as well as he was telling, whatever things come to a contact, okay, even if it is a, your fingerprint also, there is a trace, okay. Now, yeah, that's what this was showing in the slide. Lockhart principle states that once a contact is made between two surfaces, the traces will be occurring, okay. And can you go to next slide, sir? Hello. Yeah. Okay. Now we are having crime scene organizations or crime laboratory. That is, since 1960s, the organization of the crime scene laboratories has been started up. Okay. Now you'll be thinking like, what are crime scene laboratories? Why it is so? Why the crime scene lab should be there? Why the crime scene lab should be there? Since 1960s, the demand came like, whenever there is a crime had happened, there should be a proof. Yes or no? Means whenever there a crime happened, you should solve that as a forensic scientist. Yes or no? So for that, we are having, n number of forensic laboratories now. And why we need a lab? Why we need a crime lab? More number of crimes are happening day by day, more drug related crime or n number of crimes, digital related crime. Now, criminals are becoming so much proficient. So same as we also want to become, or we also want to get developed. Because of that, what is happening? The crime scene laboratories has been made. Okay. And then why the crime laboratories? For each and every department of forensic science, that is, as I told before, forensic science is a branch which covers what? Forensic biology, forensic physics, forensic chemistry, everything will be there. Okay. Everything, n number of criteria are there for forensic science. Because whenever, see, for example, if you're learning chemistry, there will be coming in the case of drug. Yes or no, the drug. What is a drug? Drug is an illegal substance and which can be legalized. Yes or no? So it can correlated with the case of forensic science. Okay, same way, another units are there. For example, can you change the slide, sir? Next, sir, next. Yeah. So within this, you can see, previous one. Yeah, thank you. So within this, we can see we have n number of units in an forensic science or crime scene lab. As I told before, biology lab is there. Firearm is related with ballistics. So ballistics lab is the documentation, question documentation, that is forgery. Whenever you are putting your friend's signature, your teacher will be scolding you, na? Like, say for example, if you are told to read this passage and come for tomorrow as a homework in school days and put your parents' sign, we are students, so what we will do, we'll put our sign. Okay, or we'll put our father's sign. We will put our father's sign. So this is known as what? Forgery. Doing or pretending to be someone to put someone's signature or disguising something, that we can find it out using n number of methods. So that is what document unit or question documentation unit. And other than that, we are having forensic photography. This is one of the interesting field. Okay, what is forensic photography now? Forensic photography is completely related with forensic units only. Mainly
Ma'am, we are unable to hear you. Are you still there?
Sorry for getting disconnected, students. Back. So here we are having. Now can you hear me?
Yes,
Hello.
yes.
So now we are having forensic photography unit, right? Forensic photography unit is completely based upon forensic science, not a normal photography. So here we are having, whenever a crime scene has been happened, the crime scene photographer will be, going there, and they'll be he or she'll be taking the n number of capturing n number of images, even if it is, whatever things has been happened in that crime scene. That department is known as what? Forensic photography. Okay. So can you go for next slide? Now, next we are having physical science unit, that or biological unit. Same thing, this actually, sorry guys, because I'm not having net net issue over here because of that. Okay. So now we'll go with the biological unit. So same as I told before, forensic photography unit or biological unit, we have another sort of biological unit which deals with the plant as well as animal. Whenever you are getting any kind of plant tissues or plant as an evidence, what will you do? You will go for the biological unit. Okay. Same as physical unit, you have what? You will go for any other things. Say, for example, you'll go for blood, you'll go for if you're getting knife. Knife is there, if you're getting knife in a crime scene or any kind of lipstick or any kind of physical evidences. Okay, it will come under physical unit. Okay, next, and other than that, we know that the most important. Can you go for next slide? Next slide. Yeah, next we are having the another one known as toxicological unit. As we know that the toxicological unit is nothing but it will be completely based upon drugs. Drugs are something which are illegal, which I told, as well as do you really think nowadays drugs are becoming more and more crucial? Cases are coming for drugs because see, whenever people want to gain money, and there are why people are committing crime. It's not only because they had to commit crime. No, there are n number of environmental factors, students, by which a criminal is been committing a crime. So you will be thinking now, what is that reason? Yes or no? So reason is nothing but see, environmental factors can be, see, if say for example, if a person is becoming a criminal, is it necessary like his or her parent or family, he's having a criminal background or he's coming from a criminal family? No, nothing like that, because see, it's all it depends upon the environmental factors and number of environmental factors by which a person is becoming or person became as a criminal. Okay, now we are having next one as fingerprint unit. So that's very interesting. What is fingerprint? Fingerprints are impressions which has been formed. Okay, we all have fingerprints, and fingerprints are, kindly note down students, fingerprints are unique. Okay, and now fingerprint evidences are very crucial in the case of crime scene because it is unique. I have a fingerprint, each and every individual in this world is having a unique fingerprint. Okay, and now is polygraph unit. Before coming to that, I'll be telling you about the forensic psychology. We all are learning psychology. We all have psychological, studies in our, PUC, means your 11th standard, 12th standard, your, BSc whole and BSc, MSc, everyone learned. But what is forensic psychology? Forensic psychology is nothing but it deals with forensical aspects or criminological aspects. Okay. See, in the case of psychology or forensic psychology, whenever a person, that's what I told, whenever a person had committed a crime, okay, whenever he or she had committed a crime, it doesn't mean that he is from a criminal background. It can be peer pressure, it can be unemploymental status, it can be anything. So, and it can be, whenever you harm me, I'll harm you. That sort of policies can be also there. Okay. So there can be n number of factors. So whenever a person commits a crime, we as a forensic psychologist will go and we'll examine him or her. Yes or no? So how we are going to examine? We'll be asking about his or her, family background. We will ask him like how he became a criminal. What is the, what is the reason why you committed this crime? So there will be n number of factors why a person had committed a crime. If it's so, what sort of test? If a person had committed a crime, he's not telling. Okay, he's not telling like I had done this crime. No, he n number of times we as a forensic scientist we asked why, how, when, where. This for this question, how for this questions he haven't given any reply. Now what is the what is the next step? We are having a psychological test that is known as polygraph test or lie detecting test. Means obviously no criminal will be coming and telling, yeah, I had done a crime. No, we have to tell n number of times like what, like why you had committed this crime. If he or she is not telling, next step is polygraph test. Now you'll be thinking, what is polygraph test? It's a lie detecting test. Means it will detect your lie. Whatever lies you're telling, it gonna detect your lie. Okay, that is what polygraph test. So could you please go for next slide? Thank you, sir. Yeah, now we are having voice print analysis. This is very interesting unit because see, we all are having good voices and everything. We'll prank our friends, right? We'll prank our friends like what to tell. I had committed like what to tell. We'll be pranking our friends in n number of ways, right? For n number of reasons or n number of issues, everything we are gonna prank our friends. So why pranking is happening, how the prank is going on, we'll change our voice to voice as another voice, right? We'll be pranking like we'll sometimes we'll increase our pitch or sometimes we'll decrease our pitch, right? So this is what voice print analysis is. If I'm calling you as if I'm calling another person as hello, my name is this thing, I'm changing my mind, changing my voice, okay, and I'm speaking and I'm telling like if you are giving me 1 lakh rupees, if you're giving me 1 lakh rupees, I'll give you a child, otherwise your child will be killed. This sort of threatening voice messages or voice calls are coming in a number of cases. This has been happened. So this is what you're pranking yourself. You're pretending to be another person and you're talking to him or her, yes or no? So after some days, if the voice analysis has been happened, what we are going to try to find it out, the voice, whose voice was that, we'll try to analyze using different types of tools. Are there children tools are something like for say for example, as I told before, the crime scene labs are there, right? Forensic crime labs are there. For what purpose is there? It's for finding how the crime had been occurred in an evidence or in a proof manner, yes or no? Same way in digital or cyber crime, we are having tools. What are tools? Tools are the applications, okay? Same way in digital forensics, we are having voice print analysis tool. We have Pratt. Pratt is an example for this thing, okay? So what we'll do, we will start to analyze the voices, okay? Whose voice was that? How he had done, why, what made him to change that voice? Okay, what made him to, prank, say for example, okay? Now, can you change next slide, sir? Yeah, thank you. Now, next one is we are having is expert witness. Who is an expert witness? Who is having a particular skill or knowledge in that particular field. Yes or no? Section 45 of IEA. What is IEA? Indian Evidence Act. So, section 45 of IEA states that about expert witness means who is having, and whoever can be an expert. If you have that knowledge, if you have that skill, if you have that, this thing in your career, that means you are an expert in that field. Okay, so you will be called into the court for the expert witness, okay, which indicate about section 45 of IEA. Okay, now, so next slide please. Now, okay, now we had learned about forensic science, we had learned lab, everything we had learned. Now, if you're becoming a forensic scientist, what is your function or what you're gonna do? First of all, if you're becoming a forensic scientist in your future, you have to be trained yourself. This is the main point which you have to note down when you become a forensic scientist, okay? means you should be trained about yourself, what you're going to do, how you're performing, how you're going to the crime scene, how you are analyzing that. Each and everything matters within the training process. Now, as a forensic scientist, you should be confidential about yourself. Means you whenever a crime scene had been happened, obviously as a forensic scientist, you'll be going and examining. Okay, fine. Now you along with another IO. IO means investigation officers will be knowing what things has been happened over the crime scene. None of the others should be known that. Means confidentiality should be maintained. Okay, next. Now, whatever, after your training process, everything had been completed, you'll go for collection of evidences, preserving that, documentation of that. Now, after that, you will be, reporting everything with the seal, package, everything will be there, right? So you'll be reporting to the consent laboratory. Okay, this is how the forensic scientist or forensic, expert should be trained. Okay, now next one. Can you go for next slide, sir? Next slide. And coming to another forensic services, we have n number of so you can change it, sir. No issue. N number of services like that: forensic entomology, forensic cybersecurity, forensic digital evidences, then forensic pathologist, forensic odontologist, forensic engineering. N number of students. I'm telling. Believe me, you have n number of forensic platform with you. If you want to succeed, in forensic field, there are n number of platform within you. You can succeed in such a way that select your era, select your topic, select your stage, and you can excel like anything in the case of forensic science. One more thing I'll wind up in. Forensic science is a developing platform. Okay, I'm not telling it's been completely developed. It's developing. Okay, it's developing like anything. So if you want to pursue something in this field, work hard, have that fire within you, you will succeed with. Okay. And one more thing, as he told before, as just I told sir before, I'm specialized in cybersecurity and digital forensic science. So even a small, introduction for this, as cybersecurity is been, emerging like anything now for every sectors, for every companies, for every cooperating companies, everything we need forensic cyber security analyst and digital analyst. So if you're really interested, you can apply for that. You can learn. You have courses like EC Council, Coursera, LinkedIn. They are providing n number of courses. Do that. Get the certificate. You will shine like anything. Okay. So this is a thing how how you can succeed in your platform called forensic science. Why I told about cyber is because it's my field, and cyber, we know that like n number of students are asking me like, ma'am, we should need the this thing Java, or we need programming language. Actually, students, we need programming language. It will be super better if you have programming language, but see, it will be fine if you don't know, if you're going to study, if you're in the basic thing, it's fine. Try to do lots of courses, you have a number of platform with you, try to utilize that. Okay. And next and another thing is about forensic psychology. Another emerging field is about digital forensic and digital forensic science is what to tell. Forensic psychology is another field. I'm doing the research on cyber psychology or digital psychology. Within this, so can you go for digital forensics? Yeah, fine, sir, fine. Yeah. So before going to digital forensic science, I'll be giving you a small gist about cyber psychology. Now it's been emerging like anything. Cyber psychology. This is another field in forensic science. Combination of cyber as well as psychology. This robotics, artificial intelligence, everything. This together combines about cyber psychology. You'll know the mindset of that, robot and how it's been working, how it's been made. Okay, everything will be jotted down within this. That is what called cyber psychology. Even this is a good field. If anyone is trying to write any research papers, articles, or anything, you can contact me. Or if you're writing, it will be well and good within this topic. It's it's been emerging. Okay. Now, digital forensic science. So, as I told before, digital forensic science is nothing but it's a platform or discipline which is completely banned with computer security or cyber security. Okay. So, what is the difference between, you'll be thinking, no, ma'am is now telling about cyber forensics and digital forensics. What is the difference? See, cyber forensics is nothing but deals with the computers, how the computers have been working, completely cyber platform. But digital is, it's a combination how, who, what, why, then, how, matlab, it is completely dealing with, collection, preserving, packaging, recovery of digital evidences. Okay. Now, in digital forensic science, we have typical investigation process. That is, first you will go to the crime scene, you will acquire that, then you'll know how to recover the evidences. Now you will start to analyze. Now you'll go and present it. So within the analysis process, that's it. That's what I told before. You have n number of tools. Okay, tools means, as I told before, it's an application used to, analyze the digital forensic science cases. Okay, now people are committing crime. Digital forensic science is not only about computers. Now it is all about another things, any digital platform in an or electronic form. Say for example, your radio, or your ATM fraud, CCTV footage. This all will comes under digital forensic science. Okay. Now you can go, sir. Go. Move, sir. Can move. Yeah. Now, what is the future in digital forensic science? As I told before, future in forensic science, digital forensic science is a platform by which you can shine like anything. Every corporate companies, every, n number of companies, Google, EY, some of my friends are working as a, digital forensic expert in Delhi. How they're getting the, they are having their own capacity, their own capability to do n number of things. I know the people who came from the biology background. Even I am from biology background. So when we have that fire in, like to learn some kind of specialization, even if it is biology, even if it is chemistry, even if it is forensic toxicology, no issue. But if you have that, try to gain yourself in any field, not only cyber, I'm telling, any field, you will succeed. Okay. So that's it all about today from me. Thank you so much, Aljos, for, and Biolix for giving me this opportunity. Thank you so much.
Thank you so much, ma'am. That was quite a wonderful lecture. I'm sure everybody will be thinking about starting a career in forensic science after hearing your lecture. Thank you so much, ma'am.
Thank you.
I now welcome Dr. Arjun Rao. Dr. Arjun Rao is an assistant professor of forensic science in the Department of Life Sciences, Christ University, Bangalore. He is a member of Raipur Institute of Forensic Science, Chhattisgarh. He received his MSc in Forensic Science from Amity University, Noida, and completed his PhD from the University of Mysore. His area of expertise includes forensic biology, forensic anthropology, forensic genetics, and wildlife forensics. He has published more than 10 international publications and 3 book chapters in reputed journals indexed by Scopus, Web of Science, Thomson, etc. He has more than 10 years of experience in the field of forensic science. And moreover, he is my former professor. I welcome you, sir.
Right. Thank you, Aljos. Am I audible?
Yes, sir. Yes, sir. You're audible.
Right. So I'll just share the screen, whether it is visible or not. Just please let me know. Is it visible?
Sir, no, sir. I cannot see it.
I had shared you the slides. Can you please make it around?
Sir, I'll just try it. Just give me 2 minutes.
Right. Bye, sir.
So can you see the slide, sir?
Yeah, that's fine.
It's full screen only, no, sir?
Yeah.
Yes.
Right.
Okay.
Right. Thank you. So may I know that who are the participants and the background of the participants? Where are they from, so that it will be easier for me to start off the lecture?
So the participants mostly are from the background of forensic science, and also we
Okay.
have a few scientific officers from different government laboratories all over India, and mainly life science backgrounds.
Right. So then we'll move ahead. Okay, so I could not make the slides what you had given me actually, the backgrounds and all. So it was a bit late of that you sent me. So then to further, we'll make it.
Sir, 1 minute, so I'll just share the different screens. I think this is not full screen.
Yeah, please.
So you missed out, sir.
Yeah. I mean, I don't go for any basic concepts of forensic science and all. I hope everybody knows about that and the branches about forensic science, how it could be dealt with, and what are the major or a minor and the different branches of forensic science. So I'll take you directly to the concepts or the actual process of work, that how actually or how basically the DNA is work, and how till now, like, might be the participants have heard about or seen the theoretical concepts by structures, that how these DNA samples will be useful in identifications, or for a two or for a three, or for an individual identifications. I'd made just small effort within my very small time. So please, next slide. I'll just.
So you can see, right?
Yeah. Could you please make it the next slide? You just go for enable editing.
Okay, so just give me a minute.
Yeah, that's fine. Full screen. Right. Thank you. Please. So am I audible to all of you?
Yes, sir, you're audible.
All right. Okay. So while starting the presentation, if any slides you have, I mean, for participants, if any queries or questions will be there, just drop a question in the dialog box so that we can take one or two questions at the end of the point. So everybody knows, okay, everybody knows that how the DNA is work, and in the different aspects of science, either it may be a life science, it can be criminal justice systems, and the types of DNA markers, how they will work. Theoretical concepts might be probably everybody knows, but what will happen when we will go with the practical issues and all? Okay, there is two types of DNA, especially one is autosomal, that is called as a mitochondrial DNA, and the other is YSNPs, Y chromosomal single nucleotide polymorphisms, which can be useful for paternity identifications, or for paternal disputes, or paternal ancestor identifications, etc. But now, what would happen is that how to use this DNA, how to make a catalog, or how to use this biological sample to create a profiling, and how could be this evidence will be useful for making one individual's profile or a community profile. When we say community profile, that means a group of peoples or a number of peoples. Okay, it's like if 10 different peoples are there, how to make one individual's DNA profile out of it, and how that individual will be identified out of the 10 or 100 or 1,000 or a lakh, I mean, a group of people. And then after, the aim of our forensic genetics is to provide that particular dialogue, that how these biological samples will be crudely analyzed, and what is that particular arrangement in which the DNA will work. And then after moving It's not only about forensic science that DNA will be used. It can be used in a different branches of forensic science, even though it can be useful for a forensic pathology and for identifying the different diseases or a complex traits or disease characteristics, to identify or to rectify some sorts of diseases and all, and especially in cases of wildlife forensics. Now, unless to the past decade, the humans or the DNA of a human was strongly read and studied. But now in the coming trends or in the coming days, years, you'll see that how this wildlife forensics, okay, or environmental forensics, how it will be raised upon. So if the students will be participate in any of these both areas of the forensic, then it will be really feel good to all of you. Okay, and then after it comes to the human identification, either for identifying the concept of a suspect, okay, or otherwise from a crime scene navigation, or to investigate possible biological relationships. What do we understand by a possible biological relationships is that whether the unknown person or whether the unknown person is maybe a father or not, or unknown person may be a mother or not, or whether there was any biological relations between the two persons, that is a male or a female, or some other dispute type of identifications and all. Right. We'll move further. I'll just please next slide. Okay, so hopefully this everybody knows that pure mitochondrial DNA of a human structure. This human mitochondrial DNA carries nearly about 16.5 kb. When we say 16.5 kb, that means 16,569 base pairs around, okay, which is similar to a chimpanzees plus or minus 10 base pairs. To that, all the species which are coinciding with the humans, okay, either orangutans, gorillas, or some other species, there will be a plus or minus 10 base pairs changes. That means it can be a 16.4 or 16.55 some kb around in other species of Homo sapiens, right? So when we talk about the mitochondrial DNA, it is very much useful in identification of ancestry, identification in forensic cases, criminal justice trials and systems. But how could it possibly work? Okay, and what it is and how it will work basically. So can you see that this circular mitochondrial DNA is having a 3 different colors? Okay, the one is with orange color, which says that it is HVR1. The other one is HVR2, which is highlighted as yellow color. And the other part of the mitochondrial DNA, which is cream in color, which is the coding region of the mitochondrial DNA. And what is coding and non-coding? I won't go to that particular specific area, but besides, I'll tell you that we as a forensic scientist or a forensic students, we are very much enthusiastically participate in the HVR1 region and HVR2 region. It is called as a hypervariable regions. And now what is hypervariable regions and why this is consecutively used in forensic studies? So one area is that when we say hypervariable, that means many variations are there in these 2 regions. Okay, in these 2 regions. So below, I had just mentioned that what are these regions and what are the positions of the nucleotides, how it can be identified. The hypervariable 1 region will start from the 16,024 to 16,069. Whereas the hypervariable region 2, it will start from 73 to 575 base pairs. These are the 2 regions where we study about the ancestry, we study about the forensic identification, we study about the maternal ancestry, we study about the paternal ancestries and all, and how this can be identified. So next, Aljos. Yeah, fine, thank you. So these are the PCR conditions. So if anybody wants to go with some type of population genetics, some types of forensic studies, some types of forensic genetics and all, where the crude method will be used, the appropriate things and all. So this is the one where we will take the study to secondary level. Okay, so this is called as a PCR, but we theoretically understand that an amplification process of our desired DNA. Okay, so my desired DNA is a complete coding and non-coding region of mitochondrial DNA. Okay, which I had studied for my study, that is 16,000 base pairs for number of samples and all. But what we can do less is that we can go for studying the HVR1 either or HVR2 either. But in forensic concepts nowadays, the third hypervariable region is also available, that is HVR3. Okay, now what we have to do is that first go and select the region of DNA you want to study. It's not only for mitochondrial DNA, it's any part of the billions of base pairs of DNA. You can study that. You just select the area, or you just select the desired area of your DNA profile, that where you want to study. And whereas we have, I have taken the mitochondrial DNA of coding and non-coding. So this particular specific area is having nearly about 23 primers. Okay, 23 primers. That means one pair of primers carries nearly about 800 to 900 base pairs amplification. So this is the amplification process I had mentioned in the first table, that is in the orange color. The different cycles what could be run in a PCR machine, and the followed by the different cycle structure I had given. But in this, only the annealing temperature or the annealing process will only be altered. Rest of the initial or the denaturation, whereas the extension and the final one, the process will be the similar, whereas the annealing temperature will be the change for all the 23 sets of primers. And the question may arise is that why the annealing temperature may changes, because there may be the hairpin, there may be triplet G, or there may be 5 sets of C's, or there may be a number of tetranucleotides present. So for that particular presence or for that particular identification, the annealing temperatures may alter. Right. Next. Thank you. I'll just. This is about a sequencing of a PCR reaction mixture, when we could possibly amplified our PCR product or a PCR zone or a PCR region, then possibly we will need to go for a sequencing. The sequencing followed by a Sanger sequencing, whereas in which we can either use a forward set of primer or a reverse set of primers, which could easily scan nearly about 700 to 800 base pairs if the capillary covers about 48 capillaries or 96 capillaries, based upon that which size capillaries we are using. So based upon that, the sequencing areas can be covered, the sequencing base pairs can be covered up. Right. Next, these are the primers what we had chosen for our studies. Okay, so these are the universal primers, we will call it as. So now what are universal primers? Okay, universal primers are those which can be implemented for any population in any location or any specific areas in the worldwide. Okay, it is not like only for India, only for the subcontinent Asia. It is not only for Europe or for American or for sub-American something. So it is used for a worldwide population. Okay, so if you have these primers, okay, you can just go for understanding that which primer which region of the mitochondrial DNA you want to study. Then you can simply just go for 1F, 2F or 23F or some other areas of primers, and then just select and just order your primers. If you do have biological samples with you, go for your extraction of DNA and just start working on the amplification of the DNA. So in the further process, you will come to know about that which region of the DNA is amplified and what is your choice of that particular specific region. These are the 1 to 13 F and R primers. F is for forward and the R is for reverse primers. These are the 13 sets of primers. In the another, next slide, it will be there possibly about 11 sets of primers. So as I said, as I said just now, that we belongs to a forensic community students or a forensic community researchers. We just want 23F R and 24F and R primers. So if these two primers, we will go for amplification for self-study even. If we are suppose having our biological sample and we are extracted our DNA, we can simply order these two primers in a sets and all. We can amplify the forward and reverse primers with 23FR and 24FR. We'll be having our own sequences of DNA, that is hypervariable region 1 and 2, with which from international database, what is available, that is NCBI or Ensemble database tool, we can just go and put our DNA sequences in the NCBI tool, and we will let NCBI will let us know that from which part of the country or which part of the globe we belongs to. Basically, there are some calculations are there. There are some software needs to be run after the sequencing and all. This is what till now I have told you about wet lab analysis. Let me tell you about the dry lab analysis. There are two different type of analysis is there. The one what we will do with pipetting with chemicals or with the reagents and all, that particular thing is wet lab analysis. And when we will move further for the analysis part in a computer or a software or a stats or a statistical analysis, that is basically called as a dry lab analysis. Yeah, this is what I had tried to show you. Yeah, before slide. Yes, right. Thank you. So this is what I just tried to show you, that that HVR1 and HVR2. So basically, we the forensic or biologists, specifically evolutionary biologist, anthropologist, or phylogenetics purposes and all, we basically study the HVR1 and HVR2, with which the motive will be successfully accomplished. Okay, with which the motive will be completely gained, that which part or which region of that particular person belongs to. Okay, the next slide, please. Yeah, this is how you will see the sequences in your statistical softwares and all. There are some free software tools available in the database. This is one of the free software that is called as a bioedit sequence alignment, with which you can also see a trial version. You can see after installing the software, you can see from the file logo that there will be so many different free sequences available in the software, which will let you know that what are the particular sequences of. You can run with the bacterial, you can run with the human, you can run with the birds, you can run with the animals, you can run with the mouse, whatever. There are some example of sequencing files are available readily with the software, right? Prakash, close it. Door. So let me take you to there. Okay, so before 360, there is one nucleotide present over there. That is G2A alteration in all of the 4 samples studied, whereas moving to the 375, where the reference sequence, it is mentioned as T, but in the studied samples, it is C. Whereas moving further, that is 395, you can see that one T is addition over there in the second number of samples. This is called as alteration. This is called as an addition of nucleotides. Whereas in the coming 404th region of the DNA, second sample, there is a T addition over there. And coming to the 409 or 408 somewhere, it is the nucleotide change over T to C. So this is how we will identify the alignments. This is how we will see that what are the changes present in our studied samples and when compared to the reference samples. So now where the mentioned nucleotides are present, let me tell you that is called as RCRS. Okay, before a decade, it used to be called as CRS, that is Cambridge Reference Sequence. And after some minor alterations and all, it is called as a RCRS, Revised Cambridge Reference Sequence. Okay, so the first lady whose mitochondrial DNA was scanned in the Cambridge University. So that was named as RCRS or a CERS, right? Next, please. Yeah. These are the samples. Yes, thank you. These are the samples what were studied basically. Okay, so how to understand this? What are these samples? Okay, we studied for all the mitochondrial DNA samples. Okay, from one particular group of peoples, and we found that these peoples, how they are inter-correlated or how they are intra-correlated. So you can just see that some typical type of groups are there. Okay, that is meant by whatever the nucleotides or whatever the SNPs they are sharing with. So this is run by a software system. Okay, these the typical DNA tables, the data what we had received from sequencing. This we will run in a software called Ehrlichman or phylogenetics or a network tree. There are so many free softwares available in which we can run our DNA samples, and then after we will get this type of images and all. So further, please, next, Aljos. Yeah, next. One more. Yeah, before this. Yeah, right. Thank you. So one more before slide. Yeah, yes. No. Next. Yeah, right. Thank you. So you can see over here the sample numbers, that is 86, 82, and the RCRS what is mentioned as, and the 06 and 33. This belongs to one particular nomenclature. So they are having one close type of relationship. They are having one close relationship with either one of the particular community, one of the particular group, or one of the particular family, one of the particular region or a clan, you can say. Further, moving to the left hand side, there is a 60, 63, 34, 31, 18, 17. These are the one particular group of peoples who are there located. Further, 123, 02, and 5, and 61. So this is how peoples will be identified, or people will be just classified, whereas they don't have any connections with rest of the peoples. Okay, rest of the peoples, they don't have any connections with them, because these are just groups, just because they are sharing some similar type of nucleotides or similar type of polymorphisms in their DNA itself. Right. One more, I'll just next. Yeah. So on the big areas, you can see that these big area, or this particular big population, or this particular big group of peoples, they might be sharing one type of nucleotides, they might be sharing one type of nucleotide polymorphisms with them. So that's why they had categorized in a bigger or in a larger number of groups. So when we call them in a single group, there will be one or two nucleotides that they may carry among their in their DNA. But when we say in a broader aspect, then they will be just having one or two nucleotides, which will be sharing by individual beings and all. Right. So this is about what we can study in a human DNA perspectives or a human identification perspectives, and how one individual is identified, or how individual can be correlated in terms of DNA and all. Right. Please, next, Aljos. So now the similar study, okay, but in terms of wildlife or a wildlife forensics issues and all. So you can see over here, one more, Aljos, please. Yeah, right. So I had just mentioned over here, okay, what we had studied for a very lesser number of samples, which we had got. Fortunately, we just got it. Okay, in the right hand side, you can see that I had mentioned the cheetal, sambar, and the red deer. They are somehow belongs to one particular similar species, where they shares similar type of nucleotides. I just tried to show the red color dots, the red color numbers, that you can see that they are just 189, 299, 319, 329, 329, and 355. And whereas the sambar or the red deer, they will also be sharing 53, 54, 214 number of nucleotide polymorphisms. This is how species of animals or humans can be identified. But here just I tried to show that how these species are correlated. Okay, in the right hand side, you had seen that cheetal, sambar, the red deer, and the topmost area, that is barking deer. These are in one clade, one area, and whereas the buffalo and bison, they are just identified in one of the particular areas, okay, one of the clade or one of the particular group of species. Whereas to the my left, I mean, the left side, you can see that nilgai and a cow is just deliberately identified in one particular species areas. And in the left to the bottom of the side, that goat and a black buck was identified in a similar type of species, similar type of group and all, right? Next, I'll just. Yeah, I mean, if just DNA and all, if it is more, then I can, I mean, take to alternate type of small, you can say, small area of researches or like in your UG or a PG or some kind of areas you can just work on. The new trends, what it is going on related of forensic science subject, that is a nanotechnology or a droplets or a droplets. No, before that slide, please. Yeah, nanotechnology in forensic science. Okay, this image is just what do you call the next world or the next time images. The one the upside, it is given as a biological nose, and whereas the downside, it is given about the electronic nose. How this basically works, everybody knows that our biological nose will be just work with our receptors and the olfactory process, where the brain will get the signals from all of these, and then the system will be identified that whether which type of product it is. So now, similarly, we have electronic nose also, where the sensory arrays are there. It will just process the data, and it will go on for database searching that what it is basically. And then after the based upon a pattern recognition, it will let you know that what the basically either a compound or either a what do you call a material or either a fingerprint or either a chemical, either a GSR product or whatever. Okay, this is one of the areas of a work nowadays, what we can see in upcoming days and all. Next, Aljos. Yeah, where this nanotechnology or what do you call a microprocessor and everything will work to identify the explosive materials detections, to estimate the time since death, even though like the process of the postmortem or the process of the death, what will happen and how many days or how much time that since death it can be identified. In a larger group of areas, the screening of drugs can also be done to facilitate the what type of crimes and all we can identify. The microparticles are to identify the fingerprint identifications. I'll just given two or three papers in the next one. Okay, the natural materials in fingerprint detections. Nowadays, there are fewer studies were carried out in broader areas, where it has identified that whatever the black fingerprint powder we do have, whatever the fluorescent powders we do have, or the type of white powders we do have in our forensic science laboratories and all, or the worldwide data suggesting that they are just very much chemical constituent composition materials, and which are very much and high risk of either cancer or high risk of health hazards, either high risk of cancerous type of reagents and all. So this particular nanotechnology or this particular nanomaterials can be useful in fingerprint identification or similar type of identifications and all. So in the next slide. So this is one of the recent, I mean, I had gone through and seen that there are so many natural products were being used to identify the fingerprint identification, to the fingerprint detections and all. Okay, so the durian seeds were used to identify the fingerprints from the crime scene area or a crime scene place. And in some studies or in some researches, natural henna powder were used, or in some areas of studies, the coffee powder was used. And so there are so many natural materials or nanoparticles kind of things were used for the present studies and all. So you all can put a similar type of effort in your areas, that whatever the nanoparticle studies or nanomaterials which can be used for altering the present process. Okay, why and how your work will be admired, or how your work will be renowned, you need to understand. The first basic thing is that whatever the present situation is, okay, we need a change, or we need a progressive change. We need a shorter time gap. We need a natural type of materials which may not cause many type of diseases. Either it can be a skin cancer, either it can be internal organ damages. There are so many there are so many reagents which were already replaced from the biological and chemical laboratories, that were replaced with some other type of chemical constituents or biological reagents and all, just to replace. Why we are replacing? Because they are either cancerous, or they are some type of allergic disorders and all. Right. One more example is there. One more paper I had just gone through with. Yeah, this one. Okay, the Fuller's Earth, what we call it the Multani Mitti, it can also be used for identifying the latent fingerprints in crime scenes and all. So a fewer researches, okay, so use just a routine process, that how this particular trending things are going on in research areas and all, and how you can use your natural instinct and natural process to identify some such types of innovative things and all. Okay, and whatever that particular issues are there which can be replaced in the current times. Yeah, I'll just right. Thank you. Yes, I can take one or two questions, please.
So we do have a question from the participants. It's says, I really enjoyed the presentations. How can I seek scholarship to study masters in forensic science?
Yeah, I need to come back for this. Okay, there are some scholarships available, but in which university are you registered for, or in which particular university you are just going through? Okay, whether that particular university is providing any scholarships and all. There are some different types of categories or different types of areas are there in which the university will be providing you a scholarship. And if it is a state, central, or it is a private university, it needs to be understand first. Then based upon that particular university or that particular organization or that particular institute, you can let know that whether that university is giving any scholarships and all. Yeah, I'll just, am I there? I'm there. I can take one more two questions from the topic which I had done. Hello?
Yes,
Am I audible?
Yes, sir, yes, sir, we can hear you.
Yes.
So thank you so much, sir. That was a very informative lecture, especially on how DNA is related to forensic science and how it is important in the field of forensic science. Mainly when we look at forensic science, what we first think is mainly only about investigations, crime scenes, murders. But you have explained to us how genetic material, how such a small genetic material in the body contributes in the field of forensic science, and how DNA techniques is used, especially DNA fingerprinting and other various methods. Thank you so much, sir. Questions? I think, sir, because it's two separate attendees and panelists. So if anybody has any questions, please do mention in the chat box. It was a very informative lecture, sir. Thank you, sir.
All right. So I can leave now. If no questions are there, I'll leave in a minute.
We'll just wait for a minute, sir.
Yeah, I'll wait for a minute. Then if not, then I'll leave.
If anybody has any questions, please do mention in the chat box.
లెక్చర్ అవుతుంది లెక్చర్ అవుతుంది
I think nobody has any questions. By this, we end today's session. Before ending the webinar, I have a few announcements to make. The certificates and the feedback link for the webinar will be shared via mail. I hope everybody will be receiving it, and please don't forget to fill in the feedback link. Thank you so much for today's session. I would like to express my heartfelt gratitude to the today's speakers, Ms. Aishwarya MV and Dr. Arjun Rao, for taking out the time from their busy schedule to deliver the lecture today, and also the participants for the active participation. Thank you so much, guys.
Right. Thank you very much. Thank you very much.